Hello, I'm Tristan Reynolds from the University of Melbourne, and I'm going to take you through the Galaxy Training Network tutorial on sequence mapping. Now, in this tutorial, we will cover how to map sequence reads using the mapper Bowtie 2 and how to view the results using a couple of genome browser viewers. Now, read mapping is the process to align sequenced reads to a reference genome. And what happens is a mapper takes as inputs a reference genome and a set of reads with the aim to align each read in the set of reads to the reference genome while allowing for mismatches, indel, so insertions or deletions, and clipping of some short fragments on the two ends of the reads. I will be using the Galaxy Australia server to show you this tutorial. Now, the first step you'll need to do is after you log into Galaxy is create a new history in which to carry out this analysis. So we go to the history panel on the right and we use the create new history, so the plus icon, that will create a new unnamed history, which we can use the pencil icon to rename to something meaningful so you can find your history at a later point in time. I will just name mine example mapping. Now we are going to get our sample, our data for this tutorial from Zenodo, which the links to our read files are in the tutorial. So we just need to go and copy from the tutorial the two Zenodo links and go back to Galaxy. On the left, in the activity bar, we select Upload. That will bring up the Upload data window. Select Paste Fetch Data, and in the text box, paste in one URL per line our two links to the data sets we are using in this tutorial. We click Start, let the progress reach 100%, and close the upload window. We can now see in our history, our two data sets are currently in the process of being uploaded. Once they turn green, they will be uploaded and ready to use within Galaxy. Now that they have turned green, we can see both our FASTQ files are available in Galaxy for us to use. Now, Galaxy uploads data sets with the original file name from where you imported it from. And what we will now go and do is rename each of these to a shorter, simpler file names for tracking. So we go into for our first data set, we select the pencil icon to edit the attributes of our data sets, and we will change the file name from WT H3K3 K4 ME3 underscore read one, and just rename this to reads underscore one. So our first set of reads. We then select save at the bottom of the window. And we can now see our file name, our dataset name has been updated from the original file name from upload to reads underscore one. We can now do the same thing for our second dataset. So we edit attributes and we rename our second dataset to reads underscore two. So our second set of reads. We save that and we can see the reads, the dataset name has been updated to reads two. So what we have gone and done here is we've just imported into Galaxy two FASTQ files, which correspond to paired end data that we are going to be using for our analysis. And by changing the file name, the dataset names to have underscore one and underscore two, we can use this to indicate the forward and reverse set of reads in our pair. Now, I'll stop here just to go and mention that during sequencing, errors are introduced, such as incorrect nucleotides being called, sequencing errors which might bias the analysis and lead to misinterpretations of the data. And the first step in any analysis you will be carrying out will be to check the read's quality and perform any cleaning on your data. There is a dedicated quality control tutorial for dealing with sequencing data. And in this tutorial, we will not repeat those steps, but you should go and follow that tutorial to apply quality control to any real data you're using in Galaxy before going on to the next steps that we will go straight to now. Now, just a couple points before we start actually doing our mapping. So when you're doing mapping, you need your reads, as we have here, and also a reference genome. So just for a bit of background on what is a reference genome, the reference genome is, or the reference assembly, is a set of nucleic acid sequences assembled as a representative sample of a species' genetic material. As they are often assembled from the sequencing of different individuals, they do not accurately represent the set of genes of any single organism, but as a mosaic of different nucleic acid sequences from each individual. For most model organisms, there are several possible reference genomes available, such as for human, there can be the HG19 reference genome or the HG38. Uh, In our case, we are dealing with ChIP-seq data for mice, so we're going to use one of the mouse reference genomes. And in particular, for this tutorial, we're going to use the MM10 uh, reference. Now, in terms of mappers, so the actual tools that we use to go and take our reads and map them to our reference genome, there are over 60 different mappers currently available for use, and Uh, as time goes on, people will create additional ones. For this tutorial, we're going to use Bowtie 2, which is a fast and memory efficient open source tool, and it's particularly good at aligning sequence reads up to 50 to thousands of basis pairs for relatively long genomes. So to select our tool, we go over to the tool panel, which if the tool panel is not currently visible, click on tools in the activity bar and that will bring it up. And just for it to show, we click on, if it's currently visible, you click on tools, it will hide it. And then you click it again and the tool panel is now visible. In the search bar, we will search for Bowtie 2 and then click on Bowtie 2 to bring up the tool input page in our central panel. Now we're dealing with paired end reads, so we need to change the default selection of single end reads to paired end. And then we need to go and select the input FastA or FastQ file, number 1 and number 2. So that's our forward and reverse reads. So our first file, that's our forward reads. We select reads underscore 1, our first data set. And then fast for the second input reads data set, we use our reverse reads. So this will be our data set 2, our reads underscore 2, which is already selected by default. Now moving down, we don't need to turn on settings for writing unaligned or aligned reads to separate files. And we also, for this tutorial, don't want to set paired end options. However, it's always possible when you're doing your own analysis, you may want to update some of these various settings. Now, for this tutorial, we're using a inbuilt index. So Galaxy goes and has a number of inbuilt indexes already available for several model organisms. So you don't need to go and find and upload your own reference genome datasets. So we're using a built-in genome index, and we're going to select the mouse, so the MM10. So the mouse and 10, select. So that is now the reference genome that we will use when we run Bowtie 2. Moving down to select analysis mode. And We're just going to use the defaults. We're not going to change to, but there are many other settings that you can use, and depending on your analysis, you may want that. Then uh, we do want to go and turn on, save the bow tie to mapping statistics to the history. So we turn that switch and select yes. And now after we've set all those parameters, we will now select Run Tool. And this will now submit Bowtie 2 as a job on Galaxy, which will now align our two reads files to the mouse MM10 reference genome. And this can take a little bit of time, so don't be concerned if it doesn't run immediately. Once Bowtie 2 has completed running, the Bowtie 2 outputs of the alignments and the mapping stats datasets will both appear green in your history. First thing we'll do is look at the mapping stats dataset. So we can click on the name of the dataset to expand and show us some metadata about the file. And we can also use the eye icon next to the dataset name to display the contents of the file. In this case, the mapping stats is just a text file, and this provides information about the quantity of the uh, mapping. We can see how many sequences are aligned, but it does not tell us something about the quality of the data. Uh, we can see that in this case, 85% of the alignments, the reads aligned exactly one time. There were, uh, well as there were 10% that were aligned more than once. Now, these results may vary slightly compared to what you will find when you run Bowtie 2, just slight differences in the individual runs of the mapper. So if your results are not exactly the same as results I have found, or that are displayed in the tutorial, which shows that in the tutorial there were 90% of reads were aligned exactly one time, don't be concerned. Now, in terms of this 10% of reads which were aligned more than one time, what can cause this? These are called multi-mapped reads, and it can happen because of repetitions in the reference genome, as in multiple copies of a gene, for example, particularly when the reads are small, which in this case. It is difficult to decide where these sequences come from, and therefore most of the pipelines ignore them. Always check these statistics to be sure of not discarding too much information in downstream analysis. We can also see the percentage of reads which have not been mapped, which is roughly 3.5%. This is broken down into explanations of why these have not been mapped. So some of these are due to the pairs aligned concordantly 0 times. So that's due to uh, reads which have been aligned disconcordantly 1 times. There's reads which have been multi-mapped. So that's ones which have been aligned greater than 1 time in the pairs aligned 0 times concordantly or disconcordantly section of the stats file. And then there's also where one read in these pairs are mapped, but not the paired read. So that's aligned exactly 1 time in the pairs aligned 0 times concordantly or disconcordantly section. And the rest of the reads, this 1882, are just simply not aligned at all. And checking the mapping statistics is an important step to do before continuing the analysis. And there are several potential sources for errors in the mapping, which are described in the mapping tutorial. And if your mapping statistics aren't very good, then you should investigate the cause of these errors before continuing with your analysis. Next, we will inspect the alignments file. So this is our BAM file or binary alignment file. You can also have SAM files, which are uncompressed versions of these alignment mapping files. And these files store the read sequence and whether they have been aligned to the reference sequence, and if so, the position in the reference sequence to which they have been aligned. So we will again click on the eye icon next to the dataset name to display the contents of our BAM file. And the BAM or SAM file contain a bunch of different information. The files always start with with lines that begin with an at symbol, and the at symbol indicates that this is metadata or comment lines in the file. Uh, and in particular, useful and important information that is contained in these metadata lines are the chromosome name and the lengths. And so these are lines that start with an SQ symbol, so such as this line here. We go past all of these comment metadata lines. We then get to the main content of the file, and BAM files consist of a table with 11 mandatory fields, but then they can also contain a number of various optional fields as well. The 11 mandatory fields are the queue name or the query template name, a flag, the reference sequence name, the position of the leftmost basis of the mapping, the mapping quality, the cigar string, the reference name of the mate or next read, the position of the mate or the next read, the observed template length, then the segment sequence, and then the associated quality of that segment sequence. And the main difference between the this and the original FastQ input files is the BAM file now contains mapping information such as the location of the read on the chromosome, mapping quality, and the other parameters that we just discussed. Now, this goes and provides quality of the mapping within a table. And what we now want to do is summarize these mapping statistics. So we'll use another tool. So we'll go over to the tool panel and we'll search for SAM tool stats. And so this tool can be used to derive statistics from both SAM or BAM files, as they essentially the same sort of file, but the BAM is just the compressed version of a SAM file. So we'll select SAM tool stats. And for the BAM file, we're using the aligned reads, so the alignments bow tie output, which is selected by default. And that's because the selected format for the BAM file is either a SAM, BAM, or CRAM file. And within our history, we only have one BAM file currently. Then we'll go down and change the use a reference sequence from no to use a built-in uh, sequence. And we'll be using the mouse, which is populated for us by default. We do not need to set any other parameters, so we'll now run the tool. Similar to Bowtie 2, the SAM tool stats tool may take a little bit of time to run, and so you may need to wait for it to complete before you can continue. So if we look at the SAM tool's stats output, we again display with the eye icon, and we can now see the summary quality statistics for our alignment. You can see in here the number of mapped reads as 98,000. See the number of mapped and paired reads, the number of unmapped reads, so the small proportion that we saw previously, and various other statistics. Now we will move on to visualizing our mapping using a couple of different genome viewers. The first one is IGV, the Integrative Genomics Viewer, which is a high-performance visualization tool for interactive exploration of large integrated genomic data sets. And IGV requires you to have the program installed on your local machine and open before you're able to view the data from Galaxy with it. So I have already installed and opened IGV. So this is what the IGV window looks like. And so now what I will do is if we go over to our history and expand the metadata for the Bowtie 2 alignments, in the set of options at the bottom below the file, the dataset metadata, we have download. I provide details and information about our data sets, the option to rerun the tool. Then there's a little miniature graph, which is for visualizing our data set. So we click on visualize. We can then select the option to display with IGV and we click on local, and this will be display our data set using the local uh, IGV viewer that we have installed. So we click on local and now the data will be imported into IGV. And if you have more than one genome installed, loaded, then select the relevant genome. In this case, we're using the mouse MM10. We go and select a specific chromosome so as to view a select section of information because you cannot view the entire genome all in at a single time. So you can't view all chromosomes simultaneously. So we select chromosome 2 and we will zoom in on a specific section of the chromosomes specified in the tutorial. So chromosome 2 and between this set of bases. We'll now go and put that into the selecting the range we want. Select go. Wait for IGV to load the section we have selected. And now we can see our visual representation of our data. There are several pieces of information displayed within the IGV plotting area. One at the top of the central section shows the depth of coverage of the mapping. For this data set, it's fairly low because we're only using a small sample of data, but for any real data analysis, you want to make sure that the coverage is fairly high and consistent across your entire genome. And the depth indicates the number of reads which are mapped to each section of your reference genome. So the more mapped reads to a specific region, the higher the depth of coverage is. Then in below that, we can see each individual set of paired end reads. Uh, and if they're gray, then that indicates that the read matches and agrees with the reference genome. If it's colored, that is to indicate if the nucleotide differs from the reference sequence by more than 20% of the weighted read. And IGV colors the bar in proportion to the read count for each base. As you will also notice, some of our reads are white, and that is because the mapping quality is equal to 0. And so the interpretation of this mapping quality depends on the mapping aligner, as some commonly used aligners use this convention to mark a read with multiple alignments. In such a case, the read also maps to another location with equally good placement. It is also possible that the read could not be uniquely placed, but the other placements do not necessarily give equally good quality hits. And in the upper right section of IGV, there's a zoom slider. So you can zoom in and out. So the minus to zoom out and view more, a larger section of the mapping. And then we can also zoom in. And if you zoom in far enough, then you will actually go and see the unique nucleotides that are mismatched to the reference genome. As you can see here, we have in at this region, there are C's in all of the mapped reads, which is in contrast to the T nucleotide in the reference genome. So this shows a unique variation between our specific sample and our reference. Now, if you do not have access to IGV, or if you just want to view your data set in the browser rather than using any locally installed software, you can use another tool which you can actually access through Galaxy called JBrowse, which is an alternate web-based genome browser. So we can now go back into Galaxy and go into the tool panel, and we will search for JBrowse, and we want to select JBrowse, not the other the any other options that are available in Galaxy. So select JBrowse. We're going to use a built-in genome, and then we'll select in for select a reference genome MM10. Then we can move down to the JBrowse in Galaxy action, and we want to use the new JBrowse instance. Then we want to insert a track group and insert an annotation track. And this will be where we provide our uh, alignment, our BAM file. So we want to select the track type as BAM pileups. And now Galaxy will select for us our BAM, our alignment file. Then we want to turn on, select yes for the auto-generate SNP track. And we want to turn on track visibility for new users. Leave everything else as the defaults, and then we will run the tool, allow that to run and complete. Once the JBrowse job completes running, we can view it with the eye icon, and this will load the JBrowse into our central viewing panel in Galaxy. We can collapse the tools panel to provide a bit more space to work with, to interact with JBrowse. And we can select to display the bowtie 2 data, the alignments, and then also the alignment style snips, our coverage. And allow that to load. And we can now see, in addition to having the actual alignments in the upper part of the viewing window, we also now have the coverage shown at the bottom, which as we saw previously in IGV, is fairly low due to the small data sets we are working with, which allows for the processing for the tutorial to be faster than an actual real data set would be. Now, we want to go and select a nucleotide range from chromosome 2, which is given in the tutorial. And this will be the region that we zoom in on so we can actually see the data. So we first want to select the chromosome that we're going to look at, which will be chromosome 2. So we use the drop-down menu to select chromosome 2. This now limits us to just looking at chromosome 2, and we will select the range that we want to actually look at rather than the whole thing. Now, one thing to note is that the format that you select the nucleotide range in JBrowse is different to the where you select the range in IGV. If we just go back to IGV, we can see that the format is the chromosome with a colon, then the start of the range with commas in between the between thousands and millions with a dash, and then the same format for the upper end of the range. In JBrowse, we similarly start with having the chromosome and a colon, but now you need to remove the commas between the thousands and the millions, and instead of a dash, it's now two dots between the lower end and the upper end of the range. So we just copy the lower end and replace the low end and remove commas. And do the same for the upper end of our range. We now select Go. And now we can see that the display looks fairly similar to what we saw in IGV. And because of the number of reads we have, the coverage is actually still being displayed, but it's just lower down. And as we can see, the coverage looks exactly the same as it did in IGV if we zoom back out. One thing you can see while looking at the coverage are there are these teardrop shapes, and these indicate that there is a difference between the reference, and it's a location where there's a difference between the reference and the reads. And you will only see these if there are sufficient reads to indicate that there is a true difference between your mapped reads and the reference. Because if there are too few reads, then it may just be at some random other error rather than a true difference. And so we can zoom in using the magnifying glass icon. And if we go and zoom in, we can Zoom in and drag across so we can look at the same position as we saw in IGV. And once you zoom in far enough, then it will resolve and show you the actual nucleotide that is different. In this case, we can see the C's, which is different to the reference. And you also note that all of our reads are colored, either red or blue, and indicates whether or not this is the forward or reverse strand. So red is a forward strand, and blue is a reverse strand. Darker shades is when the forward or reverse strand is missing its corresponding mate. So either in the case of the dark blue, that is a reverse strand, which is missing its matching forward strand, and dark red would be a forward strand, which is missing a matching reverse strand. And so this brings us to the conclusion of the mapping tutorial. Hopefully you found this useful, and good luck in the future with all your mapping analysis. Thank you. Bye.